molto commosso nell'introdurre Maria Teresa Esposito, perché è stata, quando sono tornato dagli Stati Uniti, la mia prima tesista. E quindi e poi ha fatto il dottorato SEM, è andata a fare il suo postdoc in Inghilterra, dove ora è senior lecturer in biochemistry all'University of Surrey, a una quarantina di chilometri da Londra, come hai detto. E niente, Maria Teresa è praticamente era la base fondatrice di quello che abbiamo fatto su tutta la parte dell'osso, quindi ricordiamo anche tutte le cose che abbiamo fatto con il professore Russo, con Silvia, la base di quello screening che abbiamo fatto successivamente. E niente, sono molto felice di vedere che sta bene, e pubblica benissimo e sta facendo un lavoro molto interessante sulle leucemie mieloidi acute, su cui non dico nient'altro, così ci racconti tu quello che stai facendo.
Okay, thank you, Lucio. Thank you very much for inviting me to talk about my research, and also I want to say thank you all for being here. I'm quite emotional because I told Lucio that I think the last time I have been in this room was in 2009 for my PhD viva. So it is quite an emotional roller coaster to be back in Change. But it's so nice to see so many familiar faces, so many friends that I can see sitting here. Barbara, Felice, Silvia, Professor Salvatore, Professor Zambrano, Professor Russo, Monica, I can see there, as well as new friends and collaborators, Gabriella, Dino, and Tiziana. Yeah, I think I have mentioned all the people that I know, but it's also nice to see new people. So today I will talk you about the research I have been doing since basically leaving Napoli on acute myeloid leukemia, and on particularly on KMT2A rearranged leukemia. So for those of you that are not familiar with leukemia are blood cancers, and basically worldwide we have over 500,000 new cases of leukemia every year. So if you basically compare a drop blood of a healthy patient with blood drop of a leukemic patient, you will immediately see that in leukemic patients there are lots of leukemic cells. These in the case of acute myeloid leukemia, are basically myeloid cells, but these, although they should be progenitors of immune cells, are actually non-functional. So leukemic patients and acute myeloid leukemia patients are prone to develop immune infections. So in terms of classifications, we divide the leukemia in myeloid and lymphoblastic leukemia depending on the cell of origin. So if mutations affect myeloid stem cells, they basically give rise to myeloid leukemia. If instead the mutations affect lymphoid stem cells, these mutations will give rise to lymphoblastic leukemia. In addition to this classification, we also divide the leukemia in acute and chronic, depending on the speed of the progression of the leukemia. So typically, acute leukemia, if left untreated, would basically lead to death very quickly. Now, the leukemia I have been working on is the acute myeloid leukemia. This is the most common acute leukemia in adult, and the second most common form of acute leukemia in children. More than 3,000 people are diagnosed with acute myeloid leukemia in UK every year, which basically means 8 people every day. And this is a leukemia that affects lots of, especially eldest people. And this is quite important to know because, as we all know, life expectations is improving, which means that we potentially will deal with more acute leukemic patients in the few years. The survival of these patients has changed over the past 10-20 years. So if you basically see this part of the graph on the left, you will basically see that between 1970 and 2010, there was basically there has been a change in the prognosis of acute myeloid leukemia. So these are basically patients that are younger than 60 years old. And this change, so this improvement, has been due to the introduction of the cytogenetic classification that I will talk about in a minute, as well as the fact that these patients are eligible to chemotherapy and bone marrow transplantation. That is still the main therapy for acute myeloid leukemia patients. If you instead see this graph, you will basically see that there is basically no difference. These are the patients that are over the age of 60, which basically means that for these patients, we don't have any treatment that is actually available and effective. And this is because these patients are not eligible to bone marrow transplantation and chemotherapy. So we really need new treatments, in particular for the eldest patients. The other important aspect to consider is also the fact that many patients that are eligible to the standard treatment will basically relapse due to the expansion of leukemic stem cells. Now, to explain the role of leukemic stem cells in leukemia relapse, I would like you to focus on this cartoon. When we diagnose a patient with leukemia, in these patients, we will certainly see lots of leukemic cells that we call blast, as well as leukemic stem cells that are this one in purple. When we treat the patients with the standard treatment, we certainly kill lots of blast, but we are not killing the leukemic stem cells. These leukemic stem cells that are also very good in hiding in these patients can basically give rise to relapse. And this is basically a clinical need for the patients. So what we know about these leukemic stem cells right now is that they have this mechanism of self-renewal. That means that basically they can regenerate themselves. They can be protected from chemotherapy by the niche. They are dormant, which means that usually they are not killed by chemotherapy because basically they are not cycling, and they can also evade the immune system. So analyzing and understanding the property of leukemic stem cells is now a very important aspect of leukemia biology. The other aspect that I mentioned is the introduction of the cytogenetic classification, because basically this has certainly improved the prognosis of younger patients. Now we know that basically with the advent of the next generation sequencing, patients' genome is sequenced. And thanks to this sequencing, we have been able to to classify the patients depending on the mutations that they have. And now we basically follow this new classification of the patients, where you can basically see that patients are classified in different subgroups. We know the prevalence of this particular mutation in the patients, and depending on this on the belonging of the patients to a particular subclassification, we will basically direct them to treatment. Now, in this context, you can see here that basically we divide the patients in favorable, intermediate, and adverse group. And obviously, those that are in the adverse group are the patients with the nasty mutations that won't respond to treatment. And one in particular, that is the one that has intrigued me since starting my postdoc, is the KMT2A rearranged leukemia. KMT2A leukemia is basically a leukemia which is due to one single mutation affecting the gene KMT2A, which is a histone methyltransferase at locus 11q23. So these patients have balanced translocation, which means that a piece of a chromosome is fused to another piece of chromosome, as you can see here in this picture. In terms of epidemiology, this disease affects children and adults. And quite strangely, these mutations can affect both patients with acute myeloid leukemia as well as patients with acute lymphoblastic leukemia. Within the same patients, for example, with acute lymphoblastic leukemia, we can have a transformation of the leukemia, meaning that we have a patient with acute lymphoblastic leukemia with these mutations, we treat the patient, and then eventually the same patients will show acute myeloid leukemia. So it's basically a transformation of the leukemia. And this has important implications for treatment because the myeloid and lymphoblastic leukemia are treated in a completely different way. Under a genetic point of view, this is for me extremely interesting because this is unique, and I'll show you what I mean in a minute. And the prognosis of this leukemia is very, very poor. So basically, there are less than 20% of the patients that actually survive beyond 5 years. I say that under a genetic point of view, this is a unique leukemia because many years ago, in 2002, the group of Scott Armstrong published this paper where you can see the macro array of patients with acute lymphoblastic leukemia, acute myeloid leukemia, and MLL, which is basically the other name for KMT2A rearranged leukemia. And as you can see in this macro array, basically these patients have a unique set of genes that is overexpressed. This has been studied for many years, and basically what you see here on the right are the results of RNA sequencing that have been published in 2015, where many of the genes that were originally identified in 2002 have been validated, as well as many new genes have been identified, including these HOXA genes. Now, this is very important to know if we want to treat this disease, because we need to understand why these patients develop leukemia. So, KMT2A is an epigenetic regulator, histone methyltransferases. So, normally, during our hematopoiesis, there are genes, such as this one, HOXA9, that are highly expressed in hematopoietic stem cells and gradually lost during differentiation. So our blood cells would not basically express HOXA9. KMT2A, also known as MLL, is basically one of the regulators of the expression of HOXA genes. So during normal hematopoiesis, KMT2A will basically open the chromatin by inducing this epigenetic change, histone methylation. Now, in patients with KMT2A rearranged leukemia, these genes become overexpressed because KMT2A is basically fused to other genes, and this fusion is very sticky, meaning that basically induce the recruitment of a complex epigenetic machinery that works in concert to open the chromatin and induce the expression of genes such as HOXA9. And the overall effect of HOXA9 is basically to induce the expression of downstream genes that are basically important in the context of tumorigenesis, such as kinases, as well as anti-apoptotic protein, transcription factor MYB, JUN, as well as vimentin, that are basically also protein important for the epithelial mesenchymal transition. So this is basically the main mechanism of leukaemiogenesis by KMT2A. Now, obviously, people that study KMT2A leukemia with the aim to target this oncofusion protein and to identify new treatment for this leukemia, study them under an epigenetic point of view. So what you see here is basically the KMT2A fusion protein with some of its partner genes. And you see here that this protein can basically upregulate MYX as well as HOXA, and is the action of these downstream target genes that induce proliferative metabolism and block of differentiation and apoptosis that give rise to leukemia. Now, what you see in blue are molecules that have been developed over time as potential new targets for this leukemia. There was a lot of interest on DOT1L, and I remember very well when the paper was published on this one, because this was supposed to be the ultimate therapy for KMT2A rearranged leukemia. Unfortunately, DOT1L inhibitor that were fantastic in mice didn't last very long in clinical trials because they have a toxicity that is basically unbearable for patients. Now, the most interesting target is menin. This data, there is a clinical trial ongoing on menin. The results were shown at the ASH conference, so American Association of Hematology, in December, but we know already that patients in the clinical trial are developing genetic resistance to them. So this is to say that although there is a lot of research, and in UK there are lots of groups working on epigenetic therapy for leukemia, we still don't have a treatment for it. Now, when I basically, as Lucio said, I was basically his first student, and as part of my dissertation as well as PhD, I mostly worked on mesenchymal stem cells. And when I was a PhD student, I kind of found strange that we were already thinking of using mesenchymal stem cells for regenerative treatment, but actually we didn't know if there was any risk of these cells to becoming cancer stem cells. So I decided that I wanted to study this mechanism, so I wanted to understand what are the mechanisms that transform stem cells into cancer stem cells. And this is where I basically, when I joined as a postdoc, the group of Eric So at King's College. At King's College, we basically developed this system where we can induce the expression of oncogenes, such as the KMT2A fusion protein, in mouse hematopoietic stem cells. These cells can be replated in vitro in a semi-solid medium, and they basically will give rise to pre-leukemic stem cells. When we inject these cells into irradiated mice, the cells acquire new mutations, including epigenetic modifications, and they basically give rise to leukemic stem cells. So we basically know that we can actually treat pre-leukemic stem cells with chemotherapy, and we are able to kill them. But if you try the same treatment in leukemic stem cells, they are resistant. So this was a fantastic model to try to identify mediator of resistance of leukemic stem cells. And this is basically, by basically doing a microRNA, we identified beta-catenin as a gene that is particularly important for the transition of these cells from pre-leukemic stem cells to leukemic stem cells. So this is basically our model where we basically demonstrated that by treating the leukemic stem cells with either beta-catenin knockdown or also some inhibitors, we can revert these cells into pre-leukemic stem cells. A few years later, using the same sorts of model, we also showed that the KMT2A leukemic stem cells are particular resistant to PARP inhibitors. These are poly-ADP ribose polymerase inhibitors. These are now quite famous in the treatment of BRCA-mutated ovarian and breast cancer. And in 2015, we basically tested them in the context of leukemia, and we found that some leukemia carrying some oncofusion genes are actually quite sensitive, so they are responding well to PARP inhibitors, whereas those that express high level of this gene HOXA9 are basically resistant to them. We basically show that HOXA9 is also a gene particularly important in the context of DNA damage repair, so can basically induce the expression of genes important for homologous recombination, which is one of the pathway of DNA damage repair. And we basically show that a treatment that combines PARP inhibitors with lithium chloride, which is a GS3K inhibitor, so which is an inhibitor that can reduce the expression of HOXA9, can actually increase the survival of mice. And PARP inhibitors are now in clinical trial for AML as well. So basically this is the cartoon that we presented, where we basically say there are patients, acute myeloid leukemia patients, that are sensitive to PARP inhibitors. There are some, including the one with KMT2A fusions, that are resistant, but we can revert this mechanism of resistance by modulating HOXA9 expression. Now, time for water. So when I completed my postdoc in 2015, it was basically the time to think about, okay, what else? What else can I do, and what could be my path in the context of leukemia? And I basically realized, and I started to notice this. If you look at patients with KMT2A rearranged leukemia, these patients do not have many additional mutations in addition to the KMT2A oncofusion gene. The most common are those in red, so SHP2, CBL, RAS, and basically BRAF. What these proteins have in common is that they basically are part of the classical MAP kinases pathway that we have all studied. In addition to this, patients with KMT2A rearranged leukemia often have all these kinases, MEK1, ERK1, PDK, AKT, GSK3, overactivated. So these are not mutated, these are not necessarily overexpressed, but they are overactive, meaning that they basically function much more in these patients. So I started to think, is this just a coincidence, or is something important, something that maybe is part of the mechanism of this leukemia? And there is one single phosphatase, which is called PP2A, that is an upstream regulator of all the kinases circled. So I basically presented a project to Leukemia UK, which is a charity, with the hypothesis that PP2A inactivation could contribute to the aberrant signaling of this leukemia. And this is basically where I started my independent line of research. Now, PP2A. PP2A is a serinthionin phosphatase. It's basically responsible for over 90% of the dephosphorylation on serine and threonine residues within our cells. And the activity of this phosphatase is highly regulated. So what you see in this cartoon is basically the phosphatase with its catalytic subunit, the structural subunit A. There are several B subunits that can basically be part of this complex. And there are also several PP2A endogenous inhibitors that have been identified, including SET, PME1, CP2A, and RPP19. So what we basically started to study was the individual function of this PP2A endogenous inhibitor in the context of KMT2A rearranged leukemia, starting from SET. So this one, SET, is a PP2A endogenous inhibitor that has been shown to regulate the activity of PP2A, in particular towards the kinases AKT, beta-catenin, GSK. And apart from being shown with a functional role in cancer, this protein is also quite important in Alzheimer's disease because PP2A is also very important for regulating the phosphorylation of the protein tau. In addition, and I think this is important to know, in addition to the fact that PP2A is an endogenous inhibitor of, SET, in addition to be an endogenous inhibitor of PP2A, is also a part of a complex which is called INAT, so inhibitor of acetyltransferases. And in this context, SET protects histone and non-histone proteins, including P53, KLF5s, SP1, from acetylation. So there are a few papers that basically show that by modulating the activity of this transcription factor, SET can actually modulate their transcriptional activity. So what we basically did was this. First of all, by basically using available data set, we analyzed the expression of SET in mouse leukemic stem cells. So there are basically different models of mouse AML, which can be divided in models with high leukemic stem cells and models with low leukemic stem cells. And you can basically see that the expression of SET is higher in those with high leukemic stem cells. In human, again, this was basically by analyzing available dataset, we basically found that those patients with acute myeloid leukemia and high expression of SET do actually poorly compared to those that have low expression of SET. And then we also did some correlation analysis to understand the expression of SET in the context of the leukemic stem cells genetic signature. So what you see here is basically the correlation between SET expression and some genes that have been identified as part of the acute myeloid leukemia leukemic stem cells genetic signature, and you will basically see in red some with high correlation and in blue some with low correlation. And what we also did was correlating SET in particular with the KMT2A rearranged leukemia genetic signature. And what we found here was that SET correlates very high with OXA5, OXA9, OXA10, and MACE. These are the downstream targets of KMT2A rearranged leukemia. So this gave us a hint that SET could be quite important in the context of this leukemia. So what we did was, okay, let's see the expression of SET in acute myeloid leukemia patients. And this is basically an RNA, this heat map represents an RNA sequencing of over 250 patients with acute myeloid leukemia. The one in the box are those with KMT2A rearranged leukemia. So what you see here is that basically SET is overexpressed in any acute myeloid leukemia patients with no difference in terms of subtype. We then validated the overexpression of SET in some leukemic cell lines as well as primary patients with KMT2A rearranged. And as you can see, basically, it is quite difficult to actually see SET in a healthy bone marrow, and instead the protein SET is very well visible in the bone marrow of patients with KMT2A rearranged leukemia. We also did some fractionation between cytosol and nucleus because there were papers that demonstrated that SET is able to shuttle between cytosol and nucleus, and this was important in the context of its function. And basically what we see here is that in KMT2A rearranged leukemia, the expression of SET is predominantly in the cytosol, but not exclusive. And we also know that SET is also phosphorylated on serine residues. We did these experiments because the phosphorylation of SET on serine residues, in particular serine 9, is important for the shuttle between cytosol and nucleus. Then what we did was, okay, if SET is important in the context of this leukemia, we need to understand what happens if we knock it down. So what we did was we knocked down SET in a wide range of leukemic cell lines, some with the KMT2A rearranged and some that instead were wild type. And what you see here is that if you try to knock down SET in those cells with KMT2A rearranged, these cells do not form colonies anymore, which basically means that SET is essential for the clonogenic abilities of these cells. When I saw this data for the first time, I was kind of shocked because this is what actually we want when we do the research on leukemia, so I couldn't believe it, and basically everybody in the lab did exactly the same experiments to make sure that basically this was highly reproducible. So, although what you see here is a remarkable effect of SET in KMT2A rearranged leukemia, we were able to obtain some colonies, for example, in other leukemic cell lines without other rearrangement. So we tried to study these cells to see if they could grow normally or not. And what you see here is basically a system that we used. So in our system, the leukemic cell lines are GFP positive, and we basically have one molecule of GFP per cell, so we can basically use the GFP as a reporter of viability. So you see here that K562, which is a cell line with the BCR-able rearrangement typical of chronic myeloid leukemia, they basically have an effect when SET is knocked down, but they can still grow. And instead, in other cell lines, such as the Kasumi one, which express AML1-ETO, a non-KMT2A rearranged subtype, these cells also have quite a, they basically go in cell cycle arrest. So was kind of, okay, good. So SET knockdown kills or basically induce cell cycle arrest in these cells, but we know that we can't use RNA interference in patients. So what do? We found this drug that is FTY-720. This is a sphingosine analog that is basically approved by FDA as an immunosuppressor for treatment of multiple sclerosis, because basically inhibits the egress of lymphocytes from secondary lymphoid organs. But in addition to this mechanism, FTY-720 has also been reported to disrupt the binding between SET and PP2A. So we thought, okay, we can use this as a SET antagonist and try to see if this pharmacological treatment can actually kill the leukemic cells. So these are the results of 4 leukemic cell lines, and these are the KMT2A rearranged leukemia. And as you can see, treatment with FTY-720 induce cell cycle arrest in these cell lines. And this is basically also shown here, where we quantified the cells in the different phases of the cell cycle. It was very important to demonstrate that this phenotype was dependent on the activation of PP2A. And indeed, in these experiments, we used an inhibitor of PP2A, which is called ocadaic acid. So what you see here is that if you treat the KMT2A rearranged cells with FTY-720, you basically increase the number of apoptotic cells. But if you treat the cells with FTY-720 and ocadaic acid, you basically make the cells resistant, which basically means that this mechanism depends on PP2A activation. And we also use the genetic silencing of PP2A to basically validate the same findings. We also saw that this drug, FTY-720, could actually increase the response to standard chemotherapy daunorubicin, and we did these experiments in PDX. So PDX are patient-derived xenotransplant. So you basically take the primary samples from acute myeloid leukemia patients, you inject it into an NSG mice, and then when the mice develop leukemia, the bone marrow cells are basically considered PDX. To try to understand the molecular mechanism of this drug, we performed a phosphoproteomic analysis. And what you see here is that basically FTY-720 affects cell signaling related with cell division as well as regulation of proliferation. And this was kind of expected because this is basically what we saw in terms of phenotype of our cells. With this analysis, which is called KSEA, kinase set enrichment analysis, we could also identify the kinases that were particularly responsive to treatment with FTY-720. So you see in red those whose activity basically is increased by FTY-720, and in blue those whose activity is instead inhibited by the drug. And what these, obviously we had lots of data from this phosphoproteomic analysis, but you can see here that for both the KMT2A rearranged cell lines, we identified Aurora kinases B as well as ERK, CDK2 and CDK5 as kinases that were inhibited by FTY-720. And what these kinases have in common is basically a mechanism of regulation of MYC stability. And we basically validated this mechanism with Western blot in both cell lines that we treated with FTY-720 as well as cell lines where we could knock down SET. We then also did an RNA sequencing and in the cells that were treated with FTY-720. And what we basically saw with this volcano plot is that FTY-720 downregulates genes that are important for ribosome biogenesis and instead upregulates genes that are important for granulocyte activation, which is very compatible with a typical differentiation phenotype of leukemic stem cells. What we found very interesting is that among these genes that were basically downregulated by FTY-720, we found HOXA9 and HOXA10, that, as I said at the beginning, these are the downstream target of KMT2A fusions. So we also validated this data in the cells where SET was knocked down. And what you see here is that in KMT2A rearranged cell lines, where you knock down SET, you basically also obtain a downregulation of the expression of HOXA9 and HOXA10. And this is very specific to KMT2A rearranged leukemia because we basically couldn't observe the same sort of downregulation in leukemic cells with different oncofusion, expressing different oncofusion genes. So we are almost at the end. We tried, with the help of Gabriella, Tiziana, and Dino, to understand a little better what was the molecular mechanism linking SET to HOXA9 and HOXA10. And they did some chromatin immunoprecipitation, where they basically show that in the KMT2A rearranged leukemia, we could basically observe the enrichment of SET on the promoter of HOXA10 but not HOXA9. And this data was also supported by data that were published many years ago, where actually the expression of SET in a very simple system such as the HeLa cell line was reported to activate the expression of these genes. So, in conclusion, what we think of this data is this. So, in leukemic cells carrying the KMT2A rearranged, SET is basically overexpressed. we have seen that basically most of SET is in the cytosol, but we also have observed SET in the nucleus. And other groups have shown that the translocation of SET on this serine is actually quite important for the shuttle between nucleus and cytoplasm. When SET is overexpressed, can bind basically the PP2A, and this renders PP2A inactive. PP2A can regulate the activity of several kinases, including aurora kinases B, GSK, and ERK, and therefore by inactivating these phosphatases, SET can actually increase the activity of these kinases that contribute to the proliferation of these leukemic cells. When we treat the leukemic cells with FTY720, we have shown that there is a disruption of the binding between SET and PP2A, and I don't think I have shown this here, but we actually also see that over time, FTY720 can reduce the expression of SET in the leukemic cells, and we are trying to investigate the mechanism behind this. The overall effect is that PP2A becomes active and therefore can regulate the activity of these kinases, which are important for the stability of MYC as well as the expression of the HOX genes. And we think that this is the mechanism by which the inhibition of SET can induce cell cycle arrest in these cells. So this work has been mostly done by two people that are basically Antonella and Johanna, that at that time were PhD student and postdoc in my lab, as well as my colleague Yolanda that has generated the leukemic cells, the GFP leukemic cells. As I have said, basically part of this work has been done here in Naples by Tiziana, Gabriella, and Armando. And there are also several other people that have contributed. For example, my colleagues at the Barts Cancer Institute that have basically given access to the patient sample. Many of you might actually know Luca Cozzuto, that was also a SEM PhD student, that basically he helped us with the analysis of the RNA sequencing in the leukemic patients. And other colleagues in other university in UK that have helped with the PDX or analysis of the phosphoproteomic. And these are basically the funders, and the most important for me is obviously Leukemia UK, because they believed in this project in 2017 and they awarded me the fellowship. Thank you so much for listening. If you have any question, I'm happy to answer.
Thank you very much for this beautiful seminar, Maria Teresa, and your coming. I welcome your coming back in Italy, at least for a while, in Naples particularly. And this has been very nice seminar, very nice findings you have already done, and I hope you the best for your future. But now the seminar is open to questions. Please.
Thank you so much. First of all, thank you so much for the presentation. Could you potentially see if there is a changing in the ligandome when you treat these cells with your treatment? And eventually, if there is a change, using this information to do TCR therapy, for example?
To do?
TCR therapy.
TCR therapy.
Yeah. Because
okay.
I think, but I'm not sure, that there might be that you have some genes which are overexpressed or downregulated, which might change the repertoire which is expressed on the surface of these cells. Can they become target for a targeted therapist then, which can be either antibodies or TCR therapies maybe?
Yeah, I mean, this is certainly potential. Obviously, this is difficult to do with these models of leukemia because these are immunocompromised mice, and therefore it's quite difficult actually to understand the contribution of SET in the immune microenvironment as well as in the antigen presentation. But yeah, absolutely, would be nice to look at this avenue. Thank you.
Thank you.
Okay, Lucia is back, so you may continue your...
Okay. Other questions?
Do you know about any implication of polygon complexes in this leukemia? Because usually you have that there is a balance between MLL and polygon activity.
Yeah.
They
There
are
are
implicated. They are working in some way to make more aggressive the phenotype.
Yeah, there are, for example, there are some studies with BME1 that basically have shown that this particular leukemia is actually resistant even to the knockout of BME1. But if you knock down both BME1 and HOXA9 and then induce the expression of the oncogene, you can actually kill the leukemic cells. So it is certainly relevant in the context of this leukemia.
Thank you. Just a curiosity, I have one.
No.
Sorry, Lucia. About the OXA system, have you tried besides 9 and 10 protein also some other protein of the same? Because, for instance, we have found OXA2A as a that goes down in breast cancer very much in not never found before. We are just on the way to...
I mean, this is interesting because I think that there could be a relationship between SET and HOXA. I mean, what we have demonstrated here for the context of this leukemia is a correlation between SET, OXA5, OXA9, and OXA10. But we know that HOXA genes are also important in solid tumor, and SET is overexpressed in any type of solid tumor. So this is something that I'm really interested in study, starting from the correlation as well as actually see what is the effect of the knockdown in the context of this solid tumor.
Have you also made searching on long non-coding RNA for seeing if they have some effect on...
No, this isn't something that we have looked at.
Anyway, I have already done this question to you before, but in private, but I think I would you continue to understand what happens to this oncogene that will probably make some further contribution to the system, to understand the system.
Yeah.
If you can make some preview, we may be glad to listen to you.
Yeah, absolutely. I mean, we basically don't know if what is the role of SET in the context of the leukemia. What I mean is the natural follow-up experiment to this would be to knock out or knock down SET and then induce the expression in hematopoietic stem cells and then induce the expression of the oncogenes, transplant the cells in vivo and see if these cells can give rise to leukemia. Now, here there are two problems. First of all, the knockout of SET is embryonic lethal because basically SET is also important in the context of neurogenesis. So we could overcome this problem by using fetal liver cells. This is something that it's doable. So we can basically take fetal liver cells, transduce them with the KMT2A oncogenes, and then transplant them in vivo and see if there is development of leukemia. Or we can do this with a conditional knockout. So the conditional knockout is also available. So it's something that I have requested funding for. So fingers crossed. Yes.
A curiosity. There are a number of studies also in atherosclerosis, that's why for the field, about clonal hematopoiesis and what is its role in the development of atherosclerosis and subsequently leukemias. Do you have any data at all if there is any mutation? or those associated with clonal hematopoiesis that can actually be more frequent in your type of leukemia?
This would be really interesting to know. I mean, we know, and this is published, that basically SET is highly expressed in the hematopoietic stem cells, is also expressed in the progenitor, but its expression is basically lost during the normal differentiation, which make me think that probably this is important in the context of stem cells or hematopoietic stem cells. The conditional knockout that I was mentioning actually has no defect in hematopoiesis, which also makes me think, okay, it might actually be relevant to the leukemic stem cells, which would be good, and not to the hematopoietic stem cells. But actually, in terms of clonal hematopoiesis, I don't think there is anything published to basically where people have looked at the expression of this gene in clonal hematopoiesis, for example, to see if it is more expressed in old people rather than like in pre-clones. Pre-clones, yeah.
More expressed in older people because there are some of these mutations that gives clonal hematopoiesis clearly gives you eventually phenotypes that are more severe. So that's why.
These are the kind of things that would be more interesting to do in healthy people rather than in disease. So we should basically look at the expression. Longitudinal studies where we could just look at the problem is that it's not necessarily easy to get a bone marrow from a person that has no disease. I don't know if we will be able to see this in blood. Yeah.
Well, eventually, when they developed a high percentage of clonal hematopoiesis, you can see it in blood, but you can see the mutation. I don't know if you can actually study the expression.
Yeah.
Any more questions? None? Thank you.
Thank you.
Maria Teresa.
Thank you.